Welcome to video 10 of the Search for a Better Health series. In this video, we're going to be looking at the third line of defense. So we're going to be looking at antibodies, B cells, and T cells. Now, just a heads up, this is going to be a lot of words, a lot of new phrases, a lot of processes. So make sure that you look at the pictures and also are present in class for another explanation. So if we look at the components that make up the third line of defense, there are three which we're going to be talking about today. We have T cells and they're lymphocytes, so a form of white blood cell that mature and develop in the thymus gland. So this is why they are called the T cells. They remain inactive in the blood and the lymph until they come in contact with an antigen. B cells control the humoral or the blood response, in which B cells in the blood and lymph are activated by the presence of antigens. And antibodies are produced in the lymph nodes by B cells in response to a specific antigen. So if we look at T cells, they're produced in the bone marrow and mature in the thymus gland. At maturation, they are released into the blood, spleen, tonsils, and lymph nodes. They have specific surface receptor proteins that match with antigens and produce many clones of the cytotoxic or the killer T cells that are specific to that antigen. They control the process of cell mediated immunity, which is effective against bacteria and viruses that are inside the cells, protozoa, fungi, flatworms, and roundworms, and cancerous cells and transplanted foreign tissue. Here we have a diagram. So there are four different types of T cells which are produced by the body, and they each have a specific function. So the first are helper T cells, which are sometimes referred to as TH cells. So on the surface of these cells, they have a receptor protein that can recognize only one type of antigen. When a helper cell is activated by the presence of this antigen, it can release what we call an cytokine chemical, which is sometimes referred to as interleukin 2, which activates the cytotoxic T cells and B cells for that antigen. So then, that the helper T cells, they stimulated or they activated the cytotoxic T cells, which is the next in this diagram. And these are stimulated to produce many copies or clones of themselves when they're activated by the helper T cells. And this is when they detect cells that have displayed on their surface antigens that match their own surface receptor proteins. So then they form what we call an army, and they move to the site of infection, bind with the infected cells, and release chemicals that destroy the infected cell. We then move on to memory T cells, and these are produced at the same time as the cytotoxic T cells are multiplying. They remain in the body so that the body can respond more quickly to future invasions by the same antigen. And then we have suppressor T cells, and these are responsible for stopping the immune response when the infection has been defeated. So we mentioned that they help to achieve the process of cell-mediated immunity. And here we can see a little bit of a process diagram of how that does in fact happen. So foreign material is engulfed by macrophages, and which then will display the antigen attached to their molecules, to their MHC molecules, which we'll refer to in a later diagram. So when these macrophages move to the lymph nodes, they are inspected by the helper T cells that have the receptor that corresponds to the antigen that is being presented. So then these helper T cells activate the cloning of the cytotoxic T cells and the memory T cells. And remember, that happens at the same time. Then these cytotoxic T cells leave the lymph nodes and migrate to the infection site, where they're able to bind to the antigen on the infected cell. They then release chemicals that destroy the cell, and these also increase the inflammation and attract more macrophages to help with the process of phagocytosis. Then what can happen is that they then defeat all of the infected cells, and then they stimulate the suppressor T cells to stop the production and action of the cytotoxic C cells. And remember, at the same time, these memory T cells are being produced to prevent the body from developing the symptoms to the disease a second time. If we then look at B cells, these are produced and mature in the bone marrow. At maturation, they are released into the blood, the spleen, the tonsils, and the lymph nodes, so just like T cells, and they have an antibody on their surface for a specific antigen. They have a short life, so they're activated when they encounter an antigen, and upon activation, they form plasma cells that do, in fact, produce antibodies. B cells control the antibody mediated or humoral immunity, which can defend the body against bacteria and viruses outside the cells and toxins produced by the bacteria. These are really important in forming the antigen and antibody complex. So here we have another process diagram which can summarize the antibody mediated immunity. So at first, we always need to have the antibody, the antigen presenting B cells, and they move to the lymph nodes. And these B cells are inspected by the helper T cells that have the antigen receptor that corresponds to the antigen being presented. Then the T cells then do their job in stimulating the cloning of the B cells that are specific to the antigen being presented. So then at the same time, again, like previous, memory B cells are being cloned, and the activated B cells produce plasma cells that remain in the lymph nodes. These plasma cells secrete antigen-specific antibodies that then move through the blood and the lymph to the infected areas. The antibodies combine with the antigens to form the antigen-antibody complex that activates the pathogen or its toxin. And then the inflammatory responses again attracts phagocytes and leads to the clearing of the debris. And memory B cells can produce short term immunity or long term immunity, depending on where the memory cells remain. And that means that on re exposure, again, the body is able to secrete a large quantity of antibodies and prevent reinfection. Here we see just a quick summary of both of the jobs of the B cells and the T cells, and we can see here where they mature, when they are activated, what type of immunity they produce, and what they defend against. So when we talk about antibodies, these are proteins which are produced in response to the presence of an antigen in the body. These are produced from plasma cells when B cells are activated. And remember that B cells are activated by T cells. The formation of the antigen-antibody complex causes the deactivation of the antigen. The destruction of antibodies may be achieved by the destruction of the antigens may be achieved by immobilization, blocking and neutralizing the active binding site of the antigen, or by causing the antigen-antibody complex to come together, making them easier to eliminate by phagocytosis. So here we see that an antigen has binding sites. So the antibodies, have antigen binding sites, and see that they are the same shape as the antigen. So remember that each antibody is specific to a specific antigen. Then when they're bound, they form the antigen-antibody complex. When we talk about memory cells, these are the lymphocytes that carry receptors that recognize antigens and provide a ready defense against subsequent invasions. So the antigen may be eliminated quite rapidly the next time, and hopefully it's before any symptoms appear. Immunity might only last a short time or for the rest of the person's life, depending on the length of time that these memory cells live. Here we see another diagram which shows how B cells and T cells interact and where they are produced and what their role is together. And here we have a second diagram which you might want to refer to. So in this diagram we have what we can see up here. We have the MHC2 molecule or the MHC1 molecule here. And these are molecules that are glycoprotein molecules, and they're composed of a carbohydrate molecule and a protein molecule, and they allow for the recognition of cells from the body. And so the MH1 are present on all cells that have a nucleus and involved in the recognition of antigens by T cells. And then we can see the MH2 molecules, they're present only on B cells and macrophages and are involved in the recognition of antigens on macrophages by helper T cells and the recognition of antigens by B cells. So then we can see here how a macrophage has the the antigen inside it, and then we can see how an activated helper T cell has the receptor that processes the antigen, and then at the same time we're producing memory T cells and memory B cells to ensure that any antigens are activated and that the body is more ready for these the next time. Thank you for watching video 10. Make sure you tune in for video 11.